Hi, my dear students. Myself, Anu Shashidharan, Academic Director and Biology Faculty of Rankings Entrance Training Academy. I welcome all of you in my new video lesson. Today, I am going to talk about Cell, the Unit of Life, Part 1. It's the most important topic of class 11th. First of all, I'm going to discuss what is a cell. You are very familiar. Cell is the fundamental unit of life. Okay, all the living organisms are made up of cell. Cell is the structural and functional unit of life because the whole body of an organism is made up of cells. It is known as the fundamental unit of life because it regulates all the functions of an organism. Do you know who discovered the cell? Yes. Robert Hooke, a famous scientist, discovered a cell in 1665. The cell is actually a Latin word. It means a little room. Okay. Do you know the branch of biology dealing with the study of cells? Yes. We have learned in the first video lesson the study of cells, we can call it as cell biology or cytology. Okay. Dear students, Robert Hooke first of all identified the cell from the cork. Yes, I hope you might hear about the discovery of cell by Robert Hooke in 1665. As you know, in those days, there was no well-developed microscope. He was used one of the self-designed crude microscope. This is actually the diagrammatic representation of the microscope. And what he has done, he took a thin slice of the cork and observed under this microscope, and he has identified so many compartments. He called it as the cell. I said cell is a Latin word which means a little room. Dear students, do you know which scientist had observed the first living cell? Yes, Anton von Leeuwenhoek first saw and described a living cell. One of the most important discovery in the field of cell biology, that is the discovery of nucleus. You are very familiar, nucleus as the controlling center of a cell. Am I right? Yes. Nucleus, first of all, discovered by Robert Brown in 1831. Dear students, in this lesson, first of all, we have to discuss about cell theory. Okay? Dear students, two scientists, Matthias Schleiden and Theodor Schwann, together formulated the cell theory. First of all, Matthias Schleiden in 1838, he concluded that all plants are made of cells. Okay, he is actually a botanist. Again in 1839, another important scientist, Theodor Schwann, he concluded that all animals are made of cells. But dear students, these two scientists didn't give any idea related to the reproduction, I mean the formation of new cells or cell division. Another famous scientist named Rudolf Virchow in 1855 identified and proposed omnis cellula e cellula. Okay, he is actually a physician who did research on cancer cells and concluded omnis cellula e cellula. What is the meaning? The meaning is all cells are arising from other pre-existing cells. Dear students, based on these concepts, the modern cell theory had been arised. According to cell theory, all living things are composed of cells. The cells are the basic unit of structure and function in living things. New cells are produced from or arised from pre-existing cells. Okay, this is actually the detailed outline about the cell theory. Now you can see different types of cells in human body. See, I think most of the cells you are very familiar. The first one is the nervous cell. Second one, this is a fat cell. This is actually the bone cell, ovum, blood cells, and sperms. Look, this diagrammatic representation of all these cells, what we can understand. All these type of cells are actually differ greatly in size, shape, and function. The shape of the cells are totally different, and we can conclude that the shape of a cell is actually showing great variation with the functions they perform. Look in the first cell, look at the first cell, the nervous cell. Do you know the function of nervous cell? Yes, you are very familiar. Nervous cell is one of the uh, longest cell in human body. Its main function is that to the transfer of impulses. That's why they are having a specific longer shape. Okay, do you know the function of blood cells or especially RBC? Yes, we are very familiar. The movement, I mean the flow of RBC or red blood cells through the blood, and it can carry the oxygen to different part of the body. So that's why thus they are having a specialized structure which allow the movement through the blood vessels. Okay, so we can conclude that the shape of the cells vary with the functions they perform. You are very familiar, students, some type of cells are very small. Do you remember we have seen one of the most important smallest living cell known today? We have discussed under kingdom Monera. That is called mycoplasma. Mycoplasma, you know, it is also called PPLO. We have discussed. You remember? Pleuro pneumonia like organisms, PPLO. One of the smallest living cell. Its size is about zero point 0.3 micrometer in length. A bacteria, it's have 3 to 5 micrometer. The human RBC 7 micrometer. And do you know the largest isolated single cell? Yes, the egg of ostrich. So what I am going to convey, all these type of cells are actually showing great variation in their size and shape. Okay, and you are very familiar. The cells can be broadly classified into two categories: the prokaryotic cell and the eukaryotic cell. The bacterial cell are actually prokaryotic cell, and the cells of kingdom Protista, fungi, animals, and plants, they're having eukaryotic cell that we have discussed. Do you know the major differences between this prokaryotic cell and this eukaryotic cell? Yes, you are very familiar. In prokaryotic cell, they are not having a well-developed nucleus and nuclear membrane. But look at the eukaryotic cell, they are having a well-developed nuclear membrane. Okay, a well-developed nuclear membrane, I mean a well-developed nucleus. Another important difference, you can see that in prokaryotic cell, they are not having membrane-bound organelles. Do you know? Do you remember the name of membrane-bound organelles? Yes, you might hear different types of organelles like Golgi bodies, endoplasmic reticulum, okay, mitochondria, chloroplast, like that. Such type of membrane-bound organelles are totally absent in prokaryotic cell. But look at the eukaryotic cell, it's actually having all type of membrane bound cell organelles like mitochondria, endoplasmic reticulum, Golgi bodies, lysosome, everything is present. Okay, this is actually the major differences between prokaryotic cell and eukaryotic cell. Dear students, in this lesson, we have to discuss in detail the characteristic features, the structure of both prokaryotic cell as well as eukaryotic cell. So first of all, in this session, I am going to discuss the detailed structure of a prokaryotic cell. Dear students, this is actually the diagrammatic representation of a bacterial cell. Okay, when you consider the structure of a bacterial cell, first of all, it's actually having a cell envelope. Okay, the outermost covering we can collectively call the cell envelope. Okay, the cell envelope it's actually having three layers. The outermost layer is called the glycocalyx. Okay, the second layer is called the cell wall, and the third layer, the innermost layer, is called the cell membrane. Okay, this is actually the outermost layer we can collectively call the glycocalyx. Look, this is the glycocalyx. Dear students, this glycocalyx sometimes it may be a loose sheath-like structure. If it is a loose sheath, it is called a slime layer. What we can say? Slime layer. If it is a thick and tough, sometimes the cell envelopes may be very smooth. I mean, they may be very loose structure. If it is a loose structure, what we can say? Slime layer. While in some other cases, it may be thick and tough. If it is thick and tough, we can call it as capsule. Okay, this is something about the cell envelope. The second layer is called a cell wall. You can clearly see the cell wall over here. This is the cell wall. See, do you know the cell wall is actually composed of a particular material or a special type of material called a peptidoglycan. The cell wall is composed of a substance called a peptidoglycan. The innermost layer is called a cell membrane or plasma membrane. You are very familiar. Cell membrane is actually made up of lipids and proteins. Okay, the cell membrane of this prokaryotic cell same as that of eukaryotic cell membrane. The detailed structure of eukaryotic cell membrane we will discuss in the next video lesson. Okay, so anyway, this layer we can collectively call the plasma membrane or cell membrane. It's chemically it is composed of proteins and lipids. The detailed structure we will discuss in the next lesson. Okay, dear students, inside this cell, it's actually having cytoplasm, same as that of eukaryotic cell. The cytoplasm, it's actually having very less number of cell organelles or cytoplasmic organelles. The only one cell organelle present in prokaryotic cell, that is ribosome. Okay, you are very familiar. I hope you might hear about basically two types of ribosomes are there. 70S type of ribosomes and 80S type of ribosomes. Okay, 70S type of ribosomes actually present inside the cytoplasm of a prokaryotic cell. Okay, second one is called mesosomes. Do you know what we call the mesosomes? Look, the mesosomes are actually the plasma membrane. It's actually having some infoldings. Look, certain infoldings are there present in the plasma membrane. This is called the mesosomes. Okay, do you know the function of mesosomes? It plays an important role in the cell wall formation, DNA replication, etc. In some type of blue-green algae, this the plasma membrane having some infoldings or extensions. In such cases, in blue-green algae, this is called a chromatophores. Okay, it's maybe filled with certain substances. In blue-green algae, such type of extensions are collectively called chromatophores. Okay, then third one is that the chromatin material. This is a chromatin material means it is actually the genetic material, the DNA. You are very familiar, they are not having the nuclear membrane. Okay, the DNA or the chromatin reticulum is actually scattered in the cytoplasm. Okay, this poorly defined nuclei we can collectively call the nucleoids. Okay, this is called the nucleoids. Look, another important region, you can call it as the plasmids, plasmid DNA. Another important cytoplasmic structure is called plasmids. Do you know what you call the plasmids? Dear students, this is actually plasmid, that means extra chromosomal self-replicating double-stranded circular DNA. Okay, students, this is actually the chromosomal DNA. We can collectively call it nucleoids. This is actually, the plasmid DNA is actually extra chromosomal, circular, double-stranded, and self-replicating DNA. We can collectively call the plasmid DNA. Okay, so along with these cytoplasmic organelles, it's actually having some type of inclusion bodies. Do you know what they call the inclusion bodies? Yes, the inclusion bodies may be, you are very familiar, the reserve materials of this prokaryotic cell stored in specialized structures. Such bodies are collectively called inclusion bodies. So many examples are given in the NCERT textbook, that is glycogen granules, phosphate granules, and cyanophysin granules are the most important types of inclusion bodies. Okay, the next one, along with all these type of cytoplasmic organelles, the prokaryotic cells, it's actually having some other most important parts, that is the flagella, pili, and fimbriae. Do you know what is called flagella? Look, in this diagrammatic representation, you can see a single elongated tube-like structure. Okay, this is called a flagella. Do you know the function of flagella? Yes, it plays an important role in the process of locomotion. When you consider the structure of flagellum, it's actually having three important regions. Okay, this region is called the basal body. This is actually the hook, and this is actually the filaments. So the flagellum is actually consisting of three important regions: the filaments, hook, and basal body. Okay, along with this flagellum, they're also having other type of extensions called pili or fimbriae. Okay, do you know the difference between pili and fimbriae? Pili are actually very small surface structures. You can see very small surface structures of bacteria, but they do not play a role in motility. Okay, along with the pili, they're having some other type of bristle-like structures we can collectively call the fimbriae. Do you know the function of fimbriae? The fimbriae, it plays an important role in the process of addiction. That means to attach on a suitable substratum. Okay, that is actually the function of fimbriae. Okay, this is actually the detailed structure of a prokaryotic cell. Dear students, one more thing I have to discuss, that is the bacterial cell can be broadly classified into two categories based on a staining procedure. I mean the response of bacterium based on a staining procedure. Do you know what is a stain? I hope you might hear about a stain. Stain is actually a coloring substance we are using for observing the biological things, especially in case of observing the cells and all through the microscope. We are using the stain. Likewise, one of the famous scientists named Hans Christian Gram in 1884 has introduced a staining procedure for bacteria. Okay, in this type of staining procedure, some type of bacterium absorb the stain and becomes colorful. Such type of bacterium we can collectively call the gram positive bacteria. Okay, and some type of bacterium, they are not at all absorb the stain. As a result, it becomes colorless. Okay, such type of bacterium we can collectively called a gram-negative bacterium. Dear students, this is something about the structure of a prokaryotic cell. The detailed structure of eukaryotic cell we will discuss in the next video lesson. So I hope you have understood all the concepts. Okay, I am actually requesting to each and every one of you, you have to go through this video lesson again and again for better understanding. Thank you. Have a